today.
This is mitosis.
We're doing mitosis. I think I put on the next 3 lessons. I know mitosis won't be done in a day's time, but we need to work on this one. Too many people say mitosis is cell division, and it's not. Mitosis is just the division of the nucleus. And then the cell divides next. They always say it. If you look up on the internet, cell division, you'll see mitosis given to you. But actually, if you look at this, at the whole cycle, only this part is mitosis. This cell is just living, the cell is just living, the cell is just living, and the cell divides. But mitosis is only about the nucleus. See where it says interphase right here? Interphase. Okay. If you look at a cell under a microscope that's not dividing, like most of them will be, that is an interphase. That means between divisions. Now if you look at this circle, 3/4 of the time that cell is in, he's not dividing. And then this is the time the cell divides. But the point I want to make to you is that you have mitosis and you have interphase. And interphase is not really a phase in mitosis. Now I know you have metaphase and you have prophase and metaphase, anaphase, telophase, PMAT, prophase, metaphase, anaphase, telophase. Nowhere do I say interphase when I say the words for the phases of mitosis, because interphase is not a part of mitosis. That's just a cell living. That's between mitosis phases. And we still call it phase, and I still got to explain to people that there's not 5 phases to mitosis, there are just 4 phases. And then when the cell is finished mitosis, he's in a thing called interphase, which is just, he's just living.
Resting stage?
Huh?
Is it resting?
Could hear you.
Is it resting stage? Are they resting?
Resting, only thing he's not, he ain't resting. He's busy living, making new structures. He's not doing nothing. But he's not multiplying. I think some folks call it resting stage. I don't, because he's working hard. He's working hard to keep alive. Before this happens, I gotta double my DNA. In here, they double his DNA to get ready for the nuclear division. But the bottom line is, mitosis is not cell division. That is nuclear division. Cytokinesis cell division. And you're always going to find that mitosis is always followed by cytokinesis. Then your 2 cells, they grow bigger, grow bigger, grow bigger, grow bigger, grow bigger. And finally it's time that now they're too big, it's time to divide, make 2 little cells and do it again. So the picture I use is just many like this one. But he's just growing. He's just growing. He knows this phase is coming, and he knows I got to have a double number of chromosomes for my 2 new me's. And so right here he is making his new, that's the S phase. It's called synthesis because you're making new DNA. And then G2, which is, G2 stands for gap, not growth. It's a gap between this and this. But in G2, he's still growing, but actually his centrioles are dividing. He's getting ready to do mitosis. Over here, he just finished mitosis. He's just getting bigger and bigger and bigger, just living. And he's not resting, though. He's just getting bigger and bigger. And at some point, he starts to get pretty big, and now it's time to, I need to divide myself. I'm too fat. I cut myself in half and have half of what I was. But before I do cut myself, I got to make enough DNA for the new cell. So if I have 40 in me, I'll need 80 for my new cell, so I still get 40, and he gets 40. And that's how this thing works. So let's talk about it now. eukaryotes are the ones that have a nucleus because the stuff's in a baggie. And the baggie is called the nucleus. Prokaryotes have the stuff, but it's not in a nucleus called a baggie. And you look at a cell, like most cells you look at, you cannot see chromosomes. You see a black mass, like spaghetti, just, you know, boiled spaghetti dropped in a baggie, and there it is. It's everywhere. And you can't tell one piece of spaghetti from another one. They're all, well, that's called chromatin. If you can't tell the chromosomes, we call it chromatin. All right? And they're made of, as you know, the deoxyribonucleic acid, not ribo. And there's some proteins. If I have these 46 chromosomes, what holds them together? Does histones do? They're not free, just fall apart like actually. You couldn't undo spaghetti, right? Just make piles of spaghetti. These things put together. So they're connected by histones. So you're going to see more about it. Okay? When a cell divides, it's the nucleus that leads the way. In that nucleus, the chromatin starts getting thicker and thicker and thicker and shorter and shorter and shorter. And before long, you can see there's one and there's one and there's one. But then you said there were chromosomes. The moment you can see the individual structures, you go, oh, now I see that. Here are the chromosomes. The chromatin just getting thicker and thicker and thicker and thicker and thicker. And finally, you can start seeing 1 and 2, and they're so thick now, I can see all 46 of them. We have 46. But of my 46, half came from mama, half came from dad. And they like to line up. The one from mama that controls, let's say, the length of hair, and the one from dad that controls length of hair, they like each other. They're homologous. The one from mama that controls my eye color, and my dad eye color, they like to get together. They're homologous. The one that controls length of hair will not want to pair it with one that controls eye color, because both of them work together to give me my eye color. For we know, for example, brown eyes are dominant over blue eyes, and the dominant gene, if it's complete dominance, if I got 2 there and one's B, and this is a small b, the big B tells you brown eyes. The small b tells you the other eyes, blue eyes. Now, big B will completely dominate small b. So if I have big B, small b, I got brown eyes. If I got big B, I got brown eyes. Watch somebody get blue eyes. Little b. Well, now I have blue eyes. Now, if I marry my wife, now if I got blue eyes and they're both small b's, let me show it to you. This is so cool, and I'll use this. I can work with it. Let's say this is my wife, and let's say that's me. And my wife, 1B goes in one egg, other B goes in another egg. And my sperm gets one of my little B's, and another sperm gets a little B. Now, if my baby gets this egg and this sperm, that young is going to be this. What color eyes will he have? Brown. Can we ever get a blue-eyed child?
Yeah.
Because will she always stop me? No matter what I do, there's a big B that's always going to. Now, what if her eye color was this though? Where's that eraser at? Now, her eye color was this one.
Genetics.
I want this. What's this one? Now let's see what we have here. I'm going to erase faster if I do that. Now, in the case of, I can only produce the one kind of sperm, right?
Right.
But what can my wife produce? Two kinds of eggs. One of them has the big beak. Now, I'm still producing. I only got one of them. Could we get a blue-eyed child that way?
Yeah.
What would happen if, say, this sperm and this egg got together? What would be your gene type? Big, little. And that kid's eyes are what color?
Brown.
How about if these two happen to get together? Same thing. What happens if my sperm finds her egg, and now this youngin has what color eyes?
Blue.
Could you have blue eyes and your brother have brown eyes and y'all still be kin to each other?
Yes.
Absolutely. You don't know what kind of, as a matter of fact, if you have, what if both of y'all, what if both people, both parents, what if both of them are this? Is it possible that they could have a blue-eyed child?
Yes. 1 in 4.
It'd be 1 in 4?
So does that mean that those parents are both brown eyes, right?
They got brown eyes.
Because my sister.
But I can't tell what kind of brown that is though.
My sister and her husband are both brown eyes, and they have 2 blue-eyed children.
Then that means that both times the little genes got together by pure chance. It's pure chance.
And one brown eye.
It's pure chance.
Well, 2. 2 brown
So-
eyes, 2 blues.
If this is what they are, you said they're both. I cannot look at that person's eye and tell whether it's big B or big B small b. They just brown. I look at the kids though, and the kids tell me about the parents. So if this egg from mama found that sperm from daddy, that baby is this, right? What color eyes? Let's say this one found that from daddy. What color eyes? Brown. Let's say next time this one found that from daddy. And you still write the big one first. The only way would be for this egg to find that one, and then you have the blue-eyed baby.
So you're right.
Now, no, it's 1 chance in 4. That'd be good for the lottery. Now look at the world. What do you have more? Brown or blue eyes?
Brown.
Now, if you had a million people in here, I'd expect about 1/4 to be blue eyes, wouldn't you? I'd expect 3/4 to be brown eyes. But I wouldn't know from brown eyes which ones were homozygous. Homo means what? Same.
Same.
Or heterozygous. I wouldn't know until I did some more cross tests. And flowers can do that with flowers all the time. Where you get but flowers have what's called incomplete dominance. Like a red rose pollinates a white rose, and you get pink roses. That's all incomplete. But that's even better, because then I could. And some people, there's your brown, there's your green, and there's your blue, and there's your blue. Sometimes you will see folks whose eyes change colors. I don't know about the clothes you wear, and clothing do too. The clothing you wear, but I mean, I was born with blue eyes, and now they're green. Odds are you're just about right there. And of course, I got brown eyes. My wife's got brown eyes. Robbie's got brown eyes. Tim's got green eyes. So between me and my wife, we're both. Because if either one of us is this, is that big B, that stops it right there. Genetics is fun. Sickle cell anemia is this, because sickle cell is a recessive trait, and if both you and your wife are carriers, your husband, this means you're healthy. This means you're sickle cell. So if this company came out, that child don't have sickle cell. That child doesn't have sickle cell. That child does.
One
It's
in
genetics.
1 in 4, both of
It's a
you.
1 in 4 chance.
Yeah.
The thing about it is, these carriers don't get malaria. The heterozygous trait protects them from malaria. No protection here, no protection there. So somehow there's a reason why this would be desired. Because if you live in Panama and you have this genotype, you'd be fine. When we built the Panama Canal Zone, the folks who survived are the ones we knew were like this. Malaria killed thousands of workers. I probably wouldn't have a chance down there. I probably don't have the recessive gene.
I think
If you have 2 recessive genes, then your poor child has anemia, and nothing to do about it. I mean, we thought about gene splicing, where we go in this child here and use bacteria to, you know how you cut film and you put film in it, and it's called, well, we tried about trying to insert a gene in there. We've had not enough success to warrant putting money into doing it. So right now, all we do is treatment and blood transfusions and pray that the child doesn't suffer so much. But genetics, and I'll get into it later on more, but genetics is fascinating. I love genetics. It's so cool. Okay, let's get back here. So anyway, it is during interphase, which right here tells you it's not a part of this now. Interphase is not a part of this now. But during that interphase, that chromatin is getting thicker and thicker. It's preparing to make a copy of itself. And when it's so thick you can see individual pieces, we stop saying that's chromatin and we start saying those are chromosomes. Same thing. It's how you see it. But on the chromosomes is where you have all those genes for your eye color, your hair color, how tall you are, what kind of, if you're prone to have breast cancer, no breast cancer, all this, it's all programmed in there. And sometimes your diet is what sets it off. I mean, you can be programmed to have a condition, yet you never do the thing to have the condition come forward. So sometimes, something that if you eat wrong, you have heart disease.
You speed it up.
Or you may have been predisposed to have heart disease. Had you had a better diet, you might not have gotten it. But with your bad diet, and then with your predisposition, you're a goner. Okay? All right, now, this is really thin. It's so thin you can't even see this. And then it starts to, like, you take gum and start to rot water, like take twine, start to wad it up. And it gets a little bigger. These little big masses are called nucleosomes, and that's where you find the histones. And then they start getting thicker and thicker. And you can start to see something now. I just see a bunch of spaghetti, and it start to wrap back on itself, like your rubber band, your airplane, and you wind the pedal up, rubber band starts wrapping back on itself. It gets bigger and bigger as it wraps up, on itself. Finally, it gets so thick that you can start to see it. All of this is chromatin. I can't see these things other than just a massive thread. But I can see that bad boy. It looks like an X. And that's your chromosome. So when you can see it, we call it chromosomes. And this one came from mama. And that one came from daddy. And they work across here. Big B, small b, maybe another, and it's how they're read. Now see, these are making the proteins that make your eye color. So if you got 2 big B's, they make the protein to color your eyes brown. If you got 2 small B's, they don't make that protein, other proteins, and your eyes are colored blue. And that's genetics it's a wide open field, But this is thin. I would call this chromatin, chromosome. And I can see these things. They look like X's, really. And right there is a centromere. This is like a little rubber band that you wrap around and hold it together. Okay? Now, back up. See that chromatin right there? but that's inside it. It's just balled up so tight, you start, but that's all made of DNA. It's all DNA. Even this is DNA. But it's just so watered up, you can see the whole structure in it. All right, so the chromatin is made of DNA. And it's wrapped around those histones to make the nucleosome. It's starting to get bigger now. I still can't see it though. The electron scope, it looks like a little proton, like a little proton. And the beads would be the histones. And the connector would be a thread of chromatin. I'll tell you, they're nucleosomes, you know that though.
I have a question.
Fire away.
Is the centrum always in the middle?
In what now?
Right here in the center, is always in the middle?
It is. And these things go back and forth from chromosome. When you're getting ready to do mitosis, they get thicker and thicker and thicker and thicker, become chromosomes. When you finish, they go thinner and thinner and thinner and turn back into chromatin. You only see chromosomes at the time that the nucleus is going to divide. Because chromosomes can't line up side by side because they can't do anything but trace. They only work on you when they line up real long, and now this side matches that side. When they're all balled up, they're all a big mess. So when you're undergoing mitosis, that sucker is not controlling your hair color because they're too watered up. Okay? Same picture again. And it's getting thicker and thicker and thicker and thicker. And finally you see chromosome. Now this, I said metaphase chromosome, because this is a chromosome like in metaphase. And I'll tell you more later on about it. But this one did come from your grandparent. And this is from another grandparent. But I don't want to confuse you, because right now you follow me pretty good, so I won't take it that far right now. Let's just remember that when it's real thin and you can't determine any real structure, it's called chromatin. When it becomes so thick, you can start to see the structure, and you see the mass, then we call it chromosome. It's the same stuff. It's the same stuff. Now, from the PowerPoint you saw, the one that the guy said that it was made of ribose, he said that it's, sugar and phosphate, sugar and phosphate. That would be good. If you just hadn't called this sucker ribose, I'd been happy with it. But it's deoxyribose. And I bet you knew better. You're so busy working on detail, you forget the overall stuff. And he mentioned the bases, and I like how they're your bases, and I hope you know A stands for adenine, and T stands for thymine, and G stands for guanine, and C stands for cytosine. Hope you know also they pair up in a particular way. A's never hook to C's because you got a 3 plug in, 2, and they won't hook up. You got to be 3 to 3 or 2 to 2. Okay. But before you can ever do mitosis, you got to be real sure you have enough chromosomes to put in 2 brand new nuclei. And mitosis makes sure that happens. That each new cell gets the exact copy of its brother cells so that they are the same. This is another way of looking at the cell cycle and not so much detail. I've got colors going on here. I don't know where you jump in here. I don't know what came first, chicken or the egg. It's just you just hop in and go with it. So I'll start with a single cell. Okay, this cell, and I don't know when the first cell came from. I mean, this, chicken or the egg? Well, egg came first. Who laid the egg? Chicken. Well, where'd chicken come from? There's no when. There's a cycle. Well, let's just stop right here with gap 1. Let's say that we've got a brand new cell. He's one by himself, a little bitty cell. He just starts to grow, and he grows, and he grows, and he grows, and he grows. Now, some cells, like your brain cells, they never divide again. When you lose a brain cell, he's gone forever. Injure yourself, they don't heal. A stroke, you don't get it back. We can stop the stroke from hurting more cells, but we can't give you back the cells you've lost. So if this stroke had potential to destroy 1,000 brain cells, if I can stop it at 500, ain't that better for you?
Mm-hmm.
But if 500 been gone, I can't fix those bad boys. Now, if I do nothing, then you might lose all 1,000 of them. So people with a stroke have that thing to look forward to, that maybe I can stop the fire from spreading, so to speak. And once it spreads, that ground, you ain't got it. So some cells branch off and they never do. The brain cell you have right now, you've had it all your life. Mine are 61 years old. And I hope mine don't one day get the old folks' disease. I understand, though, that folks who teach have a rare instance out. You know why?
Use it or lose it.
So much.
Use it or lose it.
My mama had a stroke last year. We played cards with her. She can play cards. And I'm happy. Because by playing cards, what you doing? Card games, a lot of folks don't like them, but they do work the mind. They do work the mind. And like right now, we're in tax season, and I leave here, my tips tree service. My mom and myself do the taxes. I run the computer, she does it. And she's discussing, like, the 941 forms, and you got to take out .330, and I'm going, she had a stroke. But the very fact she's doing that is also exercising her mind. And you're right. If you don't use it, you will lose it. And so, but some cells, like your skin cells and your heart cells and most of your cells go this way. And now, right here at this point, they know this is coming. And it takes a long time to make enough chromosome to go into a new cell. So they start right here. They start right here making themselves their copies. Now, if I'm a cell, I got 46 chromosomes to keep me happy. I got to make 92 right for another cell and make it go. That takes a long time, so we started right here in the synthesis phase. What I just showed you in that replication comes first. They replicate each other. When you had one, you now have two. And this one goes to one cell, because I'm going to be gone. If I'm the cell, I'm going to be gone. Come down the middle, and I've now become two small cells. So right now we're busy making DNA because it takes a long time. Now gap, this is G2. This is a big cell, and his DNA is about double. But now we got to figure out that nuclear membrane. I got to get rid of it because that bag is going to interfere with the process. So the nucleus starts to the membrane starts to break down, and those centrioles I tell you about, they got to start going far apart. They got to put the chromosomes out in the middle. The cell divides out in the middle. Chromosomes got to be gone. They can't be there in the middle now. So the centrioles are doubling right here. The mitochondria are doubling their numbers enough for the new cell to have. Everything's doubling. Everything's doubling. And when everything is ready, then the nucleus divides. Two nuclei in one cell. And then the cell divides, and you right back here again. But mitosis is only the nucleus dividing. And I'll have two nuclei in one cell. And then the cell cleaves. We call that cytokinesis. And when I get done, I got two brand new cells. I'm gone. But I'm there in my two little cells. You call me the mother cell. And those are called what? Does the mother cell ever get to visit the daughter cells? Why not? Who is the daughter cells? Her. In high school, I asked that question, why can't a mother cell ever visit her daughter cells? I get a good answer too. They live too far away. They don't realize the mother cell has become the daughter. She's gone. She's no longer in existence. They cannot occupy the same place in time. And I had some. I should have posted those. I had some funny answers.
You need to find out.
I had a circle on one test. Had an R. And I told my kids that if you take the diameter, and you multiply it by pi, that will give you, okay, if you take the circumference times pi, that will give you the diameter. Divide that by 2, that will give you what?
Radius.
Radius. I had a little test, and I said R. And I said, find R. Guess what one kid did? Here it is. She found R. I I almost thought, I shared that with a teacher. That was so funny. I've seen it happen since then too. And so I have to rephrase my question to say, what is the value of R? Here it is. She did much of it right there. I found it right there. That's not what I want to know. But she did I marked it wrong with the smiley face. Okay. Now, I've told you earlier that the cell cycle is just in order. The set of events is perfect. And you have G1, which is your growth. That's your gap. This growth after you just divided. You're little bitty. You're getting bigger. And you have S phase, which is synthesis, and you're making more DNA for the upcoming division. And you have G2, where you're still growing, but now you're doubling your numbers of ribosomes. You're doubling your numbers of mitochondria. I'm making enough stuff for what's going to be 2 cells one day. And then you enter mitosis, where the nucleus divides, and then the cell divides, and you're right back then to G1. And replication, that I just taught you about, that happens in the S phase of the cell cycle. I know of no fancy name for the cell cycle, not like some big old Latin word. It's just called the cell cycle. Now maybe in 15 years there'd be a big name for it, like cytoneogenesis, something like that. But right now there's nothing except the cell cycle. But I'm kind of amazed that we haven't got a bigger name for it. And too many folks think mitosis is a cell cycle, and that's not right. Mitosis is a part. A quarter of it, right? Actually, well, it's less than a quarter of it. The interphase lasts forever, and mitosis gone. I wouldn't say a quarter. We just draw it that way.
There's a video where we watched in other biology class, but it shows the cell and it speeds it up until it starts.
It's a real cell.
It's a
time
real
lapse.
Yeah.
I can probably find that thing myself.
It's a real.
Yeah.
I think it's on YouTube.
I've seen that before. Hopping around and all of a sudden.
Yeah.
I'll try to find it for us. I'll show it the next time I come in here again. So G1, S, G2, mitosis. And mitosis itself has 4 phases. I could ask the question, name the 5 phases of the cell cycle. Then you could probably say, well, interphase, and then the 4 phases of mitosis. Then you say interphase, the big one. Then we have prophase, and metaphase, and anaphase, and telophase, and that. But that'd be awkward, though. I don't like doing it that way. Because that makes you think that mitosis is actually the whole cell. Okay?
Go back one more time.
I want to see where number 5 ends. I want to see this. Okay, I'm stop right there. I ain't going any further than 5, because when I get to 6, I think 6 is going to be. Well, 6 is short. Do I want to do 6?
No.
No, I don't. I want to come in and do 6 and roll from there. And if I think about it, Roger, I'll find that. I know what you're talking about. I've seen it before too. It's a great video. But it's just time lapse. Those things are talking around all these divides. I'll try to find it. I'm making note of myself. Now, as far as I'm concerned, we're going to meet in here initially for